All right, welcome back. Okay, so here's the image of the disc-shaped mitochondrial cristae that I mentioned that are present in the euglenozoan. So remember back to cell respiration, and our mitochondrial inner membrane has these folds called the cristae, and in the euglenozoans they have this unusual kind of disc shape. So that's what it looks like. Enough of that. Let's get to our next supergroup Archaeplastida. So let's see. Let me get so you can actually hopefully see this. Okay. So here's our Archaeplastida here, and there are a couple different groups of protists, the red algae and the green algae, and then this group also includes the land plants. But we will not be talking about the land plants in this chapter. They are going to get their own chapter all to themselves later on. So let's talk about some red algae. Oh, and by the way, all of the supergroup Archaeplastida, one thing they have in common is that their common ancestor absorbed a photosynthetic cyanobacterium through endosymbiosis that became the chloroplast, which is what holds the photosynthetic pigments. So these are all going to be photosynthesizers. Okay, so red algae gets its red color from a photosynthetic pigment called phycoerythrin. And this masks the green color, of chlorophyll. Because the red algae also have chlorophyll. It's just masked by this other pigment. And the one thing that is really an important adaptation of having this red pigment for the red algae is that it can absorb light wavelengths that penetrate deeper into the ocean. And we found red algae at over 260 meters of depth. So that's pretty far down. And so kind of one of the cool things we see with red algae is depending on their depth, it affects their color. So if you have a red algae that's at a very shallow depth, it might be less reddish and maybe even more towards green. But as you go deeper, it's more important to have that red pigment. And some have even been described as almost blackish in color. If they're depending on their depth in the water. So, most are multicellular and are what we would call seaweeds. Although there is a unicellular red algae, I'll show you. I don't have an image here, but I'll show you one in lab. So some examples would be the nori sheet that we use in seaweed. So if you like to eat sushi, you have eaten some red algae probably, because that is what it is wrapped in. And you'll notice that nori doesn't have a particularly, you wouldn't think of it as red, right? So that's an example of one that doesn't quite look as red. So the red algae have quite diverse life cycles. That typically are going to not include any kind of flagellated stage, but rather will depend on water current for fertilization. And this is sort of different than a lot of protists, so a lot of the other protist groups we talked about, they might have a flagella as an adult, or maybe, at some part of their sexual life cycle. And flagella are really handy for getting the sperm to the egg. In fact, we humans use them. They're so great. But the red algae don't. And they can get away with that because they can use water currents to move gametes around and help with fertilization. And now on to the green algae. So we're starting to get closer to things that kind of look like plants, but they're not plants. So the green algae are the closest relatives to land plants. Some people have actually even advocated the idea of including green algae into a more expanded plant kingdom. But, there's a lot of argument on that. Maybe it would happen. I don't know. But right now, they are definitely not considered plants. We divide the green algae into two groups known as the charophytes and the chlorophytes. So it is the charophytes that are actually the most closely related to plants. So these are the closest relative, and we're actually going to discuss the charophytes along with plants. That is the chapter that is going to talk about them. So right now I'm going to be talking a little bit about the chlorophytes, which are also closely related to plants, but not quite the closest relative. So the chlorophytes are extremely variable. We have some that are unicellular, some that are unicellular colonial, and some that are multicellular. So let's take a look at these. So we have a couple of situations here. So we have our Cladomonas. So this would be, in this case, an example of a unicellular. But this organism is capable of being colonial as well. So let's look at an example. So here is an example. This is Volvox, and it actually is, what you're looking at is colonies of many individual algal cells that look very much, so you would have many individuals that look very much like this, sort of colonizing together and acting as a unit, and we'll look at this in lab. And we also have some multicellular algae as well. So this is sea lettuce, which looks a lot like a plant, even though it's not, but it's a multicellular green algae. So we see all kinds of different variety in terms of structure. We also have some variety in terms of habitat. Some are freshwater, some are marine, meaning that they are in the ocean, and some are even terrestrial. The terrestrial ones are you're probably going to find in a fairly moist environment, or perhaps in a symbiotic relationship with another organism. Okay, by gum, we're at our next supergroup. See, now they just fly by. Okay, supergroup Uniconta. Okay, so this Uniconta is so here we have the supergroup right here. So it includes a whole big bunch of stuff. Some of which are protists and some of which are not protists. So we are basically going to talk about this branch, the amoebozoans. But let's look at this other branch known as the opisthocons. So the opisthocons, I'm going to write that down. Well, no, I'm not going to. It's right here. It's written on the slide. So the opisthocons include animals and fungi, and then two groups of protists that are considered the closest relatives of animals and fungi. The cyanoflagellates are the close relatives of animals, and the nuclearids are the close relatives of fungi. So hopefully this illustrates the problem with trying to say kingdom Protista, right? Some of them are actually more closely related to fungi, some are more closely related to plants, some are more closely related to animals. It's kind of a big tangling mess at the moment. The nuclearids and the cyanoflagellates, we will be talking about when we talk about, so we'll talk about cyanoflagellates when I talk about animals, and we'll talk about the nuclearids when I talk about fungi. So right now we're going to focus on this clade here that we know of as the amoebozoans. So in the amoebozoans, we're going to talk about two major groups. We're going to talk about the slime molds. I know, that sounds lovely, doesn't it? And we're going to talk about the lobaceans. Oh, all right, here's my amoebozoan. So here's an example of an amoebozoan, although there is a lot of diversity in form. All right, let's talk about the lobaceans first. Let's get a new board. So these are the things you would more classically think of as amoebas. So they are all unicellular, right? This is the thing you think of when I say amoeba. They have the what we call a lobe, or sometimes we call it a tube-shaped pseudopodia. Oh my goodness. That helps it both move and feed as well. These things are all over the place. We find them in moist soils. We find them in fresh water. We find them in marine environments. They have several ecological roles. They can prey on bacteria or other protists. Some will eat detritus. Or even be parasitic. So we're looking at a couple different ones here. So here's kind of our classic amoeba, and it's extended out these pseudopodia, these tube-like pseudopodia, in this case to engulf and digest this organism. And then we have this one that kind of looks like a light bulb a little bit. And what is going on over here is that some can make a shell, either out of sand, or they can secrete. They can secrete a shell. And that is the case here. So this shell here is made of sand grains, so it can make itself its own little house. And then finally, the slime molds. Okay, so the slime molds are convergent with the fungi because they have some similar structure, including fruiting bodies, So what do we mean by convergent? Think back to previous lessons. What we're saying is that they don't have these structures due to common descent. They're not homologous. So they superficially resemble fungi. In fact, that's why we call them the slime molds, right? Because they superficially, that is supposed to say mold. It doesn't look like the word mold. Let me write that, molds. They superficially resemble them in some ways, including these fruiting bodies you see here that are going to help with spore dispersal. And just the fact that they're using spores. But they're not fungi. They are a protist. And there are two different types of slime molds that have unique forms that we distinguish based on their sexual life cycle. So there's what we call the plasmodial slime molds. And the cellular slime molds. All right, let's talk plasmodial slime mold first. So one of the giveaways of the plasmodial slime mold, and here is what we would call the plasmodium of the plasmodial slime mold, is that they're typically brightly colored. So some kind of bright orange or bright yellow or something like that. And they form a mass called a plasmodium. Hence the name plasmodial slime mold. And what a plasmodium is, and this kind of blows my mind, it's basically one large cell with many nuclei. And it results from mitotic division that is not followed by cytokinesis. So usually, right, you do mitosis and you replicate your DNA and so you're dividing your nucleus. And then typically, in a eukaryote, that is followed directly by cytokinesis, the division of the cytoplasm. Well, in this case, we don't do that. So a plasmodium is one large cell. So this one large cell results from mitotic division that is not followed by cytokinesis. So let's say in the life cycle we're starting out with one cell, and this is an amoeba-like cell, just by the way. And it divides and it divides and it divides, and you see we have many nuclei here, but we don't actually divide the cytoplasm. So we have this big mass, this big multinucleate mass that can kind of move around its environment and feed. So we sometimes call this the feeding plasmodium. So it moves through the soil, with its pseudopodia. So this plasmodium, this feeding plasmodium, moves through the soil, which is where it lives. It's not pseudopodia, and engulfs nutrients. Via phagocytosis. So cell eating, it's engulfing it using its pseudopodia. So the way this typically works is this guy is happy, this plasmodium is pretty happy, just kind of crawling around its environment, feeding off stuff. But let's say the environment kind of gets bad. In this case, and bad might be just something like temperature change that's not favorable, or maybe it runs out of food. So we'll just say when somehow the environment gets harsh, the Plasmodium will stop growing. So it's going to stop dividing, and creates fruiting bodies. Which is what you see here. So we have our mature Plasmodium that stopped dividing through mitosis, and it sends up this structure called a sporangium. And the word sporangium translates into spore house. Because within this sporangium, we are going to undergo the process of meiosis to make spores. Okay, so we make these spores, and these spores will germinate and then can fuse with a different haploid cell in sexual reproduction. Right? That's what sexual reproduction is all about, and it creates genetic diversity. So, what you're looking at here is what we call a life cycle. That's what this circle is. And wherever you see kind of the pinkish arrows, we're saying that this is the diploid state, meaning 2 sets of chromosomes. And wherever you see blue arrows, we're saying this is the haploid state, meaning 1 set of chromosomes. So in humans, most of our cells are diploid, except for our egg and sperm cells, which are haploid. And it is those egg and sperm cells that would unite in fertilization to create a diploid zygote, and the life cycle starts again. So here, what we have going on is that if we were to start right here, we have our diploid single amoeba type cell, and it divides and divides and divides and becomes a feeding plasmodium. When the environment signals it to, it stops dividing, sends up a sporangium. Within this sporangium, diploid cells will undergo meiosis to create haploid spores. These spores will germinate, and one of 2 cell types will come out, either these amoeba type cells, or we also get a different form of cells, these flagellated cells. Either way, they're both haploid. 2 haploid cells are capable of fusing with each other in the process of fertilization and creating a new diploid generation that has genetic diversity. And that is the key. Sexual reproduction generates genetic diversity. So it's not particularly surprising that when the environment becomes unpredictable or unfavorable, it sort of kicks into sexual reproduction mode. Because this is one way that it can help ensure its survival, because maybe this particular plasmodium is not well adapted to the change in the environment, but perhaps with a little genetic diversity, some of its progeny might be. So we're going to see this a lot. Whenever the environment starts to become a little sketchy, organisms that are capable of both asexual and sexual reproduction, a sort of unpredictable or harsh environment will oftentimes kickstart the organism to do sexual reproduction. Okay, so now let's talk about the cellular slime molds. So the feeding stage of the cellular slime molds typically function as individual organisms. And again, these are amoeba type cells. But when food becomes scarce, the cells will aggregate and function as a multicellular organism. I can't spell the word scarce this morning, sorry. And this aggregation is sort of similar to a plasmodium, but the cells remain separated by a plasma membrane. So it's not like one big cell with many nuclei. It's basically a bunch of aggregated amoebas. So here's an actual image of one of these. It kind of looks like a slug a little bit, and this little aggregate will migrate around looking for food. Now, if the environment is still not fabulous, these cells will actually assemble themselves into a fruiting body as well. So they will also make a fruiting body that produces spores. And these spores can do a couple different things. We can actually get asexual reproduction, but we also can get sexual reproduction where two individual amoebas fuse and then divide to produce new genetically unique amoebas. So let's look at this for a second. So there's a couple different strategies we can have here. So if we look at this figure, one thing you're going to notice is that it's mostly haploid for its entire life cycle. And that is in contrast to the plasmodial slime mold, which is, I would say, mostly diploid. So these solitary amoebas are oftentimes haploid, and they form their aggregate, they migrate around, and they can create a fruiting structure, and we can just package haploid cells into a casing and make it a spore. So it can disperse out into its environment, and a haploid amoeba will emerge. And this haploid amoeba is perfectly capable of just keep doing the cycle and existing as just a haploid individual. But let's say that we need some genetic diversity. We also can have two haploid amoebas fuse together in fertilization to form a zygote. And this zygote is actually pretty transient in the life cycle. It just exists as one cell that will undergo meiosis quite quickly to create haploid amoebas again. And then we just keep going back in this haploid cycle. So it's kind of like this real quick little foray into diploidy just to kind of get a little genetic diversity going. And finally, the opisthecons, I mentioned already. But we have some pictures, so let's look at them. So the opisthecons as a clade include the protists they include a couple of protists I've mentioned already, and they include animals and fungi. So the nuclearids, I am having a really bad time with spelling today, I'm so sorry. Okay, that is a weird spelling, in fairness. So the nuclearids are the close relatives of fungi. And so that's this guy right here. And a group called the Cyanoflagellates are the close relatives of animals. So that is this guy right here. So, and we'll talk a little bit more about each of these groups when we actually get to that part of the book where we're talking about animals and fungi. All right, so that's it of going through the different groups. I'm going to have one more video, I know, where we're going to talk about some sexual reproduction strategies in Protists, and then we'll be wrapping this chapter up. So, yes, see you soon.